Okay. Okay, welcome everyone. Thank you all for joining us today. We've been doing the webinars for quite a while now, and I'm so pleased and excited to have Gareth and Sophie with us today from Charles River. I'm going to do a short introduction, and then we'll go straight into the presentation. So behind every successful drug discovery project is a set of reliable, well-optimized assays. At Charles River, the discovery teams at Chesterford Park and Harlow develop over 100 custom in vitro assays each year to support everything from routine profiling to high-throughput screening. In this webinar, Gareth and Anne-Sophie will share how automation, particularly the Certus Flex dispenser, has become a game changer in their laboratories. From speeding up assay development to saving reagent costs and improving data quality, they'll show us how the right tools make a huge difference. Whether you're deep into screening or just starting to explore automation, we hope you'll leave with practical ideas to bring back to your own laboratories. So let me introduce the speakers. So we have Gareth Langley. Gareth is a research leader at Charles River. He leads the small molecule drug discovery programs and champions smarter ways of working through automation and design of experiments. With a background in biochemistry and enzymology, he's passionate about building robust assays that move projects forward. Before joining CRL, Gareth worked on the Enable project and completed his PhD at the University of Oxford. We also have Anne-Sophie Hoevernagel. Anne-Sophie is a research leader in the HTS and HIT ID group. With over 15 years in drug discovery, she's worked across a wide range of disease areas, developing and running both biochemical and cell-based assays using automated systems. Before joining Charles River, she was part of the screening team at Galapagos. I'd like to also introduce myself. I'm Sarah Chowdhury. I'm part of the N-Nano team in Switzerland. And as I've mentioned, we're very pleased to have this joint collaboration with Charles River. So over to you, Gareth and Anne-Sophie.
Hey, brilliant. Thank you, Sarah, for the kind introduction. I'm just going to share out a screen, so hopefully you'll be able to see those slides, and then I'll hand over to Anne-Sophie to get us going.
Yes, thank you very much, Sarah, for the introduction, and then thank you to everyone for joining this webinar. So today with Gareth, we're going to talk about harnessing modern laboratory automation to deliver faster, data-driven in vitro assay development, validation, and robust screening. So as a summary, we will start with a small introduction about Charles River, and then we will talk about our liquid handling requirement and needs. And then Gareth will continue with the overview of the Certus use at CRL, and then we will finalize the presentation with 2 case studies in biology. So first, introduction to Charles River. So Charles River is a worldwide based company, and more specifically based also into UK, as you can see on the map. We have 2 key sites for drug discovery capabilities, and those sites are the one which are the closest to London. So we have Chesterford Research Park and Harlow. Both sites are count approximately 400 scientific staff with an average of 15 years of industry experience. So Charles River is a contract research organization with integrated in vivo and in vitro capabilities, and we're working with clients like academia, pharmaceutical company, biotech, and we help them to accelerate drug discovery from target identification to preclinical development. And when we look a bit closer at the hit identification phase, we see that we have conducted more than 300 high-throughput screening campaigns for more than 100 clients in a decade. So which means that we're doing approximately 30 to 40 HTS campaigns per year, and then in different formats. So it could be in 384 or 1536 format with an average size screen of 250 compounds. We can screen our compounds. We have several libraries that are available at Charles River, but we can also screen client compound or a combination of both. And the balance for assay development, so between like new assay development and an assay transfer, is quite balanced. We offer biochemical and cellular hit identification capabilities, both molecular target-based and phenotypic across our site, but we have some sites which have more core specialism. So a bit more about Charles River. So as I said, Charles River is a contract research organization with a true collaborative partnership. So we will bring strong project leadership with a wide team, really to help you achieving your goal, but also to achieve your goal, but also to complement what you already have. So it will be really an extension of your team. We will screen then in-house compound library, as I mentioned before, or your own supplied library, thanks to our compound management group, which is present at Charles River here and more specifically at Chesterford Park. We have also in-house computational chemistry that will help you to select the right library to put against your target. And then we have also a major toolbox of hit ID strategy. So we will work alongside you to define the best approach for your needs. And then, as I mentioned before, so we will have a dedicated team, that will follow your project from hit identification through hit to lead and beyond. And Charles River is really one of these companies that will help you navigate through the complexity of drug discovery and will also help you to maximize the chance at every stage. So here is just a highlight of our tracking records. So as you can see on the slide, quite a lot have happened. So we have more than 100 preclinical candidates that are delivered through our IDD programs. Then we have more than 500 patents where Charles River scientists are listed as co-inventor. And we have also more than 80% of the FDA-approved novel drug over the last 5 years that were supported by Charles River. So we're really proud of that. So now we will go on with our liquid handling requirement and needs. So what do we look for in a bulk dispenser? So we have asked these are the question, and then we were wondering what do we would like. So I think the principal thing that really popped up was really the liquid classes. We really wanted a bulk dispenser that could dispense like different kind of solvents, DMSO or aqueous solution, but wanted also to have the speed and the precision of it. We wanted also something which is also DOE compatible, so meaning that we really want to be able to dispense cells or bits, and to do that, for example, in a hood. So the portability is also really important. We also wanted something that the consumable costs were acceptable and with a long lifetime, and which was also compatible for 1536, and also integration for automation in a later stage on the platform, for example. So we have looked at that, and the Certus was really one of the best, because, for example, we have also quite a lot of combi here at Charles River, but the tips are not really flexible to really dispense on the plate. I mean, everywhere on the plate, and also it can only dispense some aqueous solution. So we're really happy that you could find the Certus. So if you go on to the next slide, thank you. So what do we want to dispense? As I mentioned, so we wanted really something that could dispense quite different of liquid class. So the Certus has been developed for contactless dispensing with a broad range of liquid class. So as you can see, listing on the slide, aqueous solution, DMSO, solvent, cells and protein, but also with a great accuracy. So you can see here also that we're using the Certus with the valve. The Certus is also a compressed, it's a pressure-based equipment. And so when the air comes to the liquid, then when there is no current, then the valve is closed. But when there is like a current going through the valve, then we have this part of the valve, which is the mobile anchor with the valve bore. It will be magnetically pulled by the magnetic field, and then the valve will open, and then the fluid will be released into the plate. So that's a really precise valve as well for the dispensing of the liquid. So this is our Certus in our lab. So as you can see, we have here the plate where we put the plate holder. Then we have the head here. So this is a straight head but we have also an angle head. We have the 8 different valves, so 4 on this side and then 4 on the other side. And then this can be, so the valve can be directly combined with like or a syringe, or combined for bulk dispensing, for example, to a bottle or a 50 ml Falcon tubes. So how we achieve it? So we're using this microvalve, this SMD 300GC microvalve. So as you can see, we have a broad range that are available at Geiger, but we are using the one which are in black, so the 0.15, 0.2, and 0.3. And we have chosen this following the volume that we wanted to dispense, as well as what we wanted to dispense. So for example, the 0.15 is really good for aqueous, and DMSO is really recommended for those liquid class. But for example, with the 0.2, you can start to dispense cells and protein, and with the 0.3 even more, because you can dispense like beads and viscous solution. So that's really helpful. We have one of each, and that's really handy for us to switch between those. Concerning the supply kits, so we have also different kind of configuration. So as I mentioned before, you can connect the Certus to the syringe or to a tube or to the bottle, and then you can have different connection. So it can go to just like one valve, but you can also connect like 2 valves out of, for example, the tube or the bottles. So generally, we use a syringe for really like precious reagent and where we don't have a lot of spare liquid. But for example, for really like high throughput screening campaign, we will use like preferably the tubes and the bottles, more for bulk dispensing. So concerning the valve cleaning station, we have also invested in that because the valve has a sub-micrometer diameter nozzle, which is really small, and it will get at a certain time clogged with the solution that we are using. So we're trying to apply good habits there, meaning that we are filtering the solution that we have, and we are also, once we have used the valve, we are directly putting them in the cleaning station to make sure that there is no left solution inside. So this cleaning station, we have written 2 main protocol on that, and just to standardize the cleaning, and that really improve the quality of the cleaning of the valve, and they will also long last. So it's a really also walkaway equipment, so we're really glad to have that. And then, so Gareth, I will hand over to Gareth with the overview of the Certus use at Charles River.
Thanks, Anne-Sophie. So we've just had kind of a whistle-stop tour of some of the things that we've got at Charles River and what we've got in terms of the Certus. So I guess the next obvious question is, what do we use it for, and kind of where has it impacted our workflows? So I've put this into two buckets here, sort of around assay development and validation, moving into more sort of routine profiling and HTS. So in the assay development validation space, which also encompasses assay transfers from third parties, this is where we are seeing huge benefits in terms of being able to go straight into final formats of 1536 wells, using designer experiments, so producing complex experimental designs and plating patterns, using the CSV import into Certus control. We've got an example of that in one of our case studies later. Complex matrix experiments, which I guess is a continuation on a theme, again, making the most of that ability to use a CSV import. Then moving more into then in the routine profiling phase, making sure we've got low volume and 1536 well bulk dispensing on sub-microliters scale, where we would predominantly use the Certus control software to control the instrument. Within that, we then got some other bits and pieces that sort of span the two. We've been starting to recently, and we're going to talk a little about it in a moment, about using the Certus to run time courses and using layers and timers to help really add value and add precision to the experimentation that we're completing, and as well a little bit about working with what I would call awkward reagents, and that might be because they're non-aqueous, but the other piece in there is that some of the reagents that are required for assay development and screening are very, very expensive, and so that things like dead volumes become a significant concern when we start thinking about how do we efficiently enact a screening campaign or conduct routine screening when the reagent's expensive, we don't want to be losing too much by the way of dead volumes at the beginning and end of runs. One example of how this impacted our workflow is that one of our junior scientists gave us the feedback that the Certis was easy to program and set up using the Certis control software, and they had a matrix experiment with different additives and different conditions. They turned a whole day at the bench into half a day and 3 plates into 1 plate, and it removes some of the complexity and maybe stress involved with plating up things in a complex manner, where you're putting some elements into some wells and some into another. And we're able to pre-program in a couple of incubation steps as well. So they were just able to set it all up, press go, walk away. And that really made what was potentially quite a daunting piece of work a lot easier for that junior member of staff. And we'll start to see that come as an emerging trend as we start to discuss some of the more complex things that we might be looking to do as we start introducing multifactorial design of experiments later on. One of the things that happens at conferences is you get chatting to people, and this is a question that came to us following SLAS in San Diego this year. Yes, it was in San Diego. And colleagues at other companies came back, asked us how were we using the Certis in terms of the valves and the fluids, and we thought, well, if somebody else in the industry is asking us that, then I'm pretty sure that the sort of people that are joining us today are going to be interested as well. So we've sort of mentioned already that we're mainly using the 3 sort of valve types that allow us to go down to about 100 nanoliters, focusing in on mainly aqueous sort of buffer, like sort of the biology solutions up to sort of cells, things with high serum, maybe high detergent as well. So the majority of it is the use of the 0.15 and 0.2 microvalves. We've got four or more of each of those, so we're able to use those quite effectively. And we find that those have been very, very good for sort of the vast majority of the things we do in the biochemical and basic cellular space. What we're looking to do is expand that in the cellular space moving forward. And we like to push our automation at CRL. We like to see how far we can push it. And so we've been using the 0.2 valves as well for some bead dispensing as well. So we've been successfully using those for SPA beads and alpha beads and sort of pushing it beyond maybe some of the technical specs as well. But we've also got the larger valves for larger cells. And I think the really big learning, and a thing we've sort of alluded to previously, and maybe say again now and possibly in the Q&A at the end, is that having that cleaning station available is a really valuable thing for an instrument that is at the heart of multiple things we're doing in the lab. So it's got access to lots of different users, being used on a near daily basis by multiple people. Is that having that cleaning station really helps us in sort of keeping standards high and making sure that we've made it easier for people to do the right thing at the end of the day. Simple, take the valves off, stick them in the cleaning station, walk away. They're clean for the next person. It just makes everything easier. And you think about when we go to sort of the more traditional peristaltic type systems where we're constantly running through heads time and time again, it means that we're able to increase the lifetime and make the most of the microvalves. So again, in terms of thinking about the environment and the environmental impact of the work we do as in vitro biologists, which, let's be honest, it isn't great with the amount of plastic where we use, being able to help reduce that by cleaning and having good practices it can only be a good thing for the wider environmental concerns as well. This is some of our internal data, moving on to it. So for our internal QC protocols, sub 2% CVs from a single channel at 0.5 microliter, parallel dispensing sub 2%, so performing in line with everything that's in the specifications in our hands. More importantly, outperforming the systems that we had in place before we got in the Certis. So again, it complements the automation ecosystem that we have within the discovery team here at Charles River, at Chesterford Park, and at Harlow. So it's providing and adding capabilities and providing us with options to feed in as part of wider automation strategies, as well as a standalone instrument itself. So this is one of the things we did very early on when we were test driving the D2 as well as the Certus. We've got some cells that, by admission of the assay scientist, always look a bit sick, but we dispense those manually with the Certus and with the D2, and they are different pictures, honestly. One of the challenges we set our biology team here is to work out which one's which. They couldn't really tell, and we can go through that in more detail if we want, but what we're seeing is that we're dispensing the cells nicely, we're getting good coverage, and yeah, and again, a shout out to the team at UK Robotics with the D2. I know that the team at Nano and Chris Geiger have worked with them really closely over the years. That is another instrument that we have at Charles River and has got some really excellent uses in other areas as well. So that's kind of a very brief sort of feel for the sorts of things that we are doing and using the Certus for at Charles River. Now what we're going to do is just go through a couple of case studies in a bit more detail, so you can hopefully get a better understanding of how the instrument fits within the sorts of things we're looking to do and how it can then impact on our outputs, both for us and for our clients. So I mentioned awkward assays and awkward solutions earlier, and some of the technologies that we use to assay different aspects of biology are inherently awkward. They could be expensive reagents, they could be hard to dispense, things like beads, things like antibodies. They could just be really gloopy and horrible proteins with high glycerol, because proteins can be divas at times. They can be highly sensitive to external environment, whether that be, in this case, we're thinking about is light. So one example we have is that alpha screens, alpha ELISA technologies. It's brilliant. It's a fantastic technology for looking at proximity on a molecular level. But if we think about the reagent cost, the sensitivities to the external environment, and being hard to dispense, it kind of falls in to being all three of those buckets of being quite tricky. But the upside is absolutely massive. So at that point, then we've got to work out how can we make this work. So we're asked by one of our partners to move in and transfer in an alpha ELISA assay, and the collaborators started off by saying that the assay that they were transferring in was problematic. So we want an assay to bring in that we know is going to be hard from a practical perspective, and we need to run it every single week to support a medicinal chemistry program. So that immediately, from a project lead and discipline lead's perspective, is then gets us thinking, well, how are we going to reliably and robustly screen? Because we need to have this to work every single week across the lifetime of a project. That could be anything from 6 months up to 2, 3, 4 years at times. And my philosophy on a lot of my science is to go back to first principles and work from there. And those that on the call that work with me know that I quite like writing things down on the back of a bit of paper on a whiteboard and going back to the first principles. And I think that philosophy here is kind of the answer to that hypothetical question there, is understand the assay and automate it. And so our assay transfer approach is based upon design of experiments. And again, more details about how that fits in with the CertiFlex is coming up in our second case study. But what we want to do is run an assessment of that assay up front, push it to its limits, understand the things that make it tick. If somebody transfers an assay in, I kind of want to break it. I want to take a data-driven approach and understand how robust is it. Is there a particular component that if you take it out or don't have enough of it, that completely kills the signal? What things do I need to know? Because that might help me if there's a problem later on, that I know where to go looking on with it. Again, we'll talk a little bit more DOE in a minute, but design of experiments, DOE, the ability of the Certus to basically plate up a unique condition in every single well of a plate. It's incredibly powerful in that early stage, whether that's assay development or assay transfer. We'll think about the standard pharmacology pieces, knowing benchmarking our known tools, particularly when we're talking about the longer sort of hit to lead optimization programs. We're not just starting everything at hit ID here. We're picking up, we're transferring in, transferring out all phases along that preclinical pathway. I think we'll probably go on to a bit more now, and this case study is to talk about screening routinely. So we want to make sure that we're not relying on one person with green fingers in the lab. As brilliant as those people are, everybody needs to be able to do it. People need to be able to pick up the assay and be responsive, so that we can keep the project moving at all times. And this is something that really came into particular focus when we were having to change the workforce working patterns during the height of the pandemic. And those sort of ideas and philosophies really do carry through to today. And the other advantage, if we've got expensive reagents, we've got really precious reagents. I mean, some of us have spent many years of our life trying to produce a protein, particularly in academic environments. So if you've got a protein that's like that, you don't want to be wasting that. So reducing the assay volumes, reducing the dead volumes, if we are able to do that in a sensible manner, that can only be good for the project. It allows us batches to last longer, means that we get more consistency across data. It means that it's a win-win scenario. And a side-by-side comparison of just progressing out with a multi-channel, electronic multi-channel versus the Certus saw us an increase in the Z prime from 0.6 to 0.8. And that wasn't from changing the similar background because it was the same assay. That was from reducing variability, so making sure that we've got more consistent, better dispensing into that plate. Probably says a little bit about how badly my pipetting skills are, but that's the main focus on it. So this is a lot of detail. We're not going to go through it in a huge amount of detail now, but to give you a flavour of this is all our routine screening is all run through the Certus, and we're using the Certus control software to enable that. And the advantage of that is that we can keep reagents in the dark as and when we don't. We turn the lights off. We can build a box fort around things to stop light leaking in. If it was really that light sensitive, we've got the ability to clean those valves. It helps us be more consistent. We've been able to implement this assay with now three or four different users, and it's working consistently every week, and it's allowed us to keep the medicinal chemistry program moving and allows us to keep things going. So this is potentially also where the Certus is very powerful, but also for these sort of smaller routine pieces that you could also consider something like the D2 as an option as well. But there's more we can do. So we've got our 384 well primary assay that supports our medicinal chemistry, supports our workhorse for the start of the project. But what else can we do with this? So I'm always thinking about how we can push things forward, how we can add more value to the project. So the easy thing was we can miniaturize it and use it for additional hit finding. That was sort of the obvious thing we could do pretty straightforward, and that's something that we've done and pursued in the background. But the other thing we were thinking about was how could we leverage the automation to maybe add more value to the project, and rather than just running a program using the standard metric of we'll get some IC50s or EC50s depending on the assay in question, can we start to interrogate the fundamental biochemistry of our assay a bit more and start to look at things like kinetics, understand key variables that are going to maybe help us work out what our focus needs to be from a medicinal chemistry perspective, and start to tease out and use a data-driven approach to accelerate our drug development timelines. And so this is where we've gone with it. We've used timers within the software to conduct time courses, and in this case we're looking at the kinetic parameters Ki and Kinact. We could also think about other types of assay formats like a jump dilution assay, or start to think about plating things up in a complex manner to enable us to do more traditional inhibition constant type experiments and start to look at mechanism of inhibition in addition. So we're starting to think and push the boundaries and just use the automation, because I spent 4 years of a PhD doing lots of enzyme kinetics and time courses and things, and these things are hard from a technical perspective. So if we're able to automate things, it reduces our error rate. It means that we're more consistent. There's less variation between operators, and we can really accurately hit those short time points. So if you've got things that are going really fast, then actually you can then start to improve your data quality and reduce error rates. So we're thinking about things like KI, K-inact. In this case, you could think about sort of some of the Krippendorf KI type experiments that we've presented in other fora previously. There's a whole bunch of different possibilities here, and the software that controls the surface and the way in which the valves work allows us to really access a lot of possibilities that you generally haven't been able to do with automation outside of perhaps very expensive solutions as well. And so that kind of summary of where we were with that initial case study is that we've incorporated the Certus Flex into our lab ecosystem, and it's changed the way we're able to think about some of those pieces around assay transfer and the difficult technologies. And the example and the second piece there about the layers and timers within the software has allowed us to think a bit differently about how we set up our experiments and maybe fully automate things that in the past have been done. Fiddly time courses, I think that, yeah, anybody who's done loads of enzyme kinetics will know that that is one of them, the trickier pieces, and it's changed the way we think about those pieces. And it's all about reducing that barrier to entry. It reduces that barrier to entry. It means that you start to think about doing the difficult but high value pieces. I'm not saying we do difficult things for difficult things' sake. I'm saying we do it because it adds significant value to the project. So it reduces that barrier to entry. It means we do what I would say the right thing and the highest value thing earlier. So we're using a more data-driven approach to our assay transfers, our assay development, our sort of follow-up assays on key pieces, how we can, the thing that's, that would be really nice if we could do that, but becomes, it would be really nice if we could do that, and let's just do it. And that piece around changing our philosophy around assay transfers, using DOE more and more, leads us nicely on to our second case study, and I'll hand back briefly to Anne-Sophie.
Yes, thank you, Gareth. So indeed, as Gareth mentioned, the design of experiment is really at the heart of our assay development approach, and that really helps us to understand more the assays, and indeed also to enter in the screening campaigns and routine screening also quicker. The design of experiment also help to reduce the downtime and also save on reagent and FTE time. So there is like a model for the DOE experiment. So first you will like predict, use a model, and also predict what your next experiment will be. So you will choose like different variable and what you want to see as a response. Like if I change the volume, does it really change the signal of a background of my assay? Or maybe if I extend the time, maybe the signal of a background and the variability would be better. So you will choose this variable and then you will specify them, and then you will design and build like a model around it. And then at the end, what you want is collect data and really analyze every different configuration that you had. But sometimes we have quite a lot of variables that we want to analyze, and then it leads to really complex design. So this cannot really be done manually. We need to have something which is automation driven for that. And that's why we're really using the Certus, and that really complements that nicely, this DOE experiment that we want to do. So we had a cellular reporter assay experiment where we had 2 parallel readouts. But in that experiment, we wanted to look at different variables like the cell seeding density, the trigger concentration, maybe the incubation time was playing also a role in that. And then we had these 2 solutions that we were adding. One was for the luminescence readout and the other one was for the absorbance. So we have created this space filling design of experiment in JUMP. So Gareth will talk about that in a few next slides. And then it was 384 runs, and each run was conducted with and without a stimulus to enable the calculation of the signal of a background. So thanks to that, we could really really shorten our timeline and really do in one experiment quite a lot of variables. So we had a CSV file that was generated with all these different volume and addition for each fluid. So for example, the cell density, as I mentioned earlier, but also the trigger and the two solutions for the readout, which lead us at the end with 768 unique wells plated in 384 well plates using the Certus Flex. And that was really handy for us. And as Gareth is a DOE super user, I hand over back to him.
Yeah, so when we set up a designed experiment, so we use JUMP as our software, it gives us a bunch of numbers that often don't really mean a lot. We've got the example on the left there is we've got some variables, we've got some outputs. How do we turn that into something that somebody in the lab can actually use? And more importantly, how do we then get that to the Certus so that we can actually make it work? What we leverage here is the ability to import CSV data into the Certus control software. So that allows us to have each well having a unique set of conditions imported. So we'll start off with building some sort of table with all the additions of the different fluids. We may need more than one stock. This can be quite a lengthy process if we're doing it manually. I'm sure that either on this call or in the follow-up that there'll be some of our colleagues at some other companies who have been looking at this problem specifically, but we'll generate the CSV file and import that into the Certus. So we'll have potentially different stocks and different things, and each fluid would then normalize within that CSV and then feed that in on top as a layer into the Certus control software. There's no reason after that you can't then start adding in extra layers after that. So we can take this CSV file, we can feed it in, and then you'll end up with some sort of plate that looks like that. Maybe not quite like that in a real assay, obviously, but that sort of piece there where you can do quite a complex design into different parts of the plate very quickly and get that into the software very, very, very efficiently. And that's been a real sort of easy way of implementing DOE. Get the fluids defined, get your volumes of addition into that CSV, bang it straight into the software, and then you are just a case of just making up the solutions and pressing go. So that's where we've really, in the first example, we were adding complexity within the software itself, and in here we're kind of doing some of the legwork offline, but then using the functionality of the software to read in those external sources of data and feed it in, and then be ready to go with quite complex experimental designs. There's no reason we couldn't have multiple layers with multiple CSVs in there as well. It's all about coming up with the best automation and the best strategy for the particular question you're trying to ask of the automation of the assay. So that process from a CRL team side looks like is that we'll start off in JUMP, generate those conditions, output that into a CSV file with the liquids, feed that into the Certus, plate out, generate the data, and then align it and feed that back into JUMP to build out and get the data analysis done, and then potentially go back to the start again, depending on where we are in the process. It may be a couple of rounds. It's all about the question that we're asking, and it's building the right assay, conducting the right experiment at the right time to allow us to progress towards our overall objectives. So in this example, I'll hand back to Anne-Sophie, where we've done the space fill, and then we start to have a look at the results.
Yeah, indeed. So we did look at the space filling in JUMP. So as you can see here, we were wondering, what was indeed really more significant for our assay. So as you can see, we have the different parameters that we have evaluated in these experiments, and we also have evaluated 2 different time points, so which was an hour stimulation versus 24-hour stimulation. And as you can see, the 1-hour stimulation is really clearly not long enough, and it has the effect of distorting our model. So meaning that when we look at the data, everything looks significant as from cell density, the volumes, or the trigger concentration. So thanks to the Certus, we had really quite a lot of data points run at 24 hours. So we could really also take it through only with the 24-hour stimulation and really analyze that for the experiment. So when we looked indeed into the JUMP software, so here you can see on the first axis that's the luminescence signal over background, and then we read out at 2 different absorbance as well. And here you have also the different parameters that we have measured. So as I mentioned earlier, we looked at different cell density. So here it was between like 10K, 50K, different also trigger concentration and different volume of the both solution that we were reading out. And what you can see, so the results were quite flat, let's say, for that experiment, but we could still have, maybe like 2 key variables that were really playing a role there. That was the cell density. So with an increase in cell density, we also saw an increase in signal over background for the luminescent assay, but as well for the absorbance one. The trigger concentration also give a key variable, but only on the luminescence-based assay. On the absorbance, this was really flat, did not give any difference, whatever concentration we were using. And then the volume of the readout solution 1, which was an absorbance 1, indeed also played a role on the absorbance readout, but not on the luminescent one. And the volume of the solution 2 give the same result for luminescence or absorbance readout. So based on those data, we decided to go for some assay conditions, so meaning an increase in cell density and also like a volume of the readout of solution 1 that was maybe in between, but still ended up with a nice signal over background at the end. So the conditions were validated, and we use that now as part of our screening cascade. So the assay is in routine bi-weekly. And we have successfully met the milestone criteria for our client and also delivered high-value chemical matter to the client. So it was really happy, and it was also in one short experiment. So the summary of this case study number 2. So it's really thanks to the searches, we really could perform a huge experiment into one single experiment because with each channel, with each valve that could really access all the wells, we could really have one combination in one well, and that really helps us to define the right combination for our assay at the end. And really this CSV import key feature really helps it all, and also the analysis that we coupled to that with the JUMP software was really helpful. So I would say that now we really adopt the DOE in our assay development and in assay validation and assay transfer. It really helps us to reduce the timelines, but we have also data-driven assay development and more robust assay at the end of the assay development. We have also put another example on the right-hand side, which looked a bit more significant to the one that we showed before. So here we also have different parameters that were measured, and sometimes you can see really that we have clear peaks that were identified and where the conditions were really clear what had the significant impact on the assays. So it was easier to choose and to go on with those conditions that also increase the signal over background at the end.
Thanks, Anne-Sophie. And I guess we're coming towards the end of the time, so just some closing thoughts and maybe summarize some of the pieces that we've discussed earlier. And the key piece is that we're data-driven in the way we do things here in the biology team at Charles River, and that generating the right data at the right time is a key part of our philosophy, and that by incorporating the Certus Flex into our automation ecosystem here at Chesterford Park and at Harlow, it's given us greater capability to generate more data in parallel. So that reduces the number of experimental cycles, which again is at the philosophy of DOE, and means that we're able to accelerate timelines. And that has the biggest impact on the ugliest, worst, most horrible protocols that you could imagine. So the things that have got big, long differentiation times, big, long incubations, if we can reduce the number of experiments, then that is going to have huge impacts on us being able to meet aggressive timelines, accelerate drug discovery from the biology team. And we can be more efficient in the way we do things. We can be more efficient in terms of reagent usage. And I'm not going to go off on one about DOE now, although I could quite easily talk about it for another hour. But things like understanding all of your interactions and understanding all of how everything fits together as a whole system, rather than just looking at one piece in isolation. And we've talked a lot about the Certus, and it's a fantastic piece of equipment, but there is, of course, not one piece of automation to rule them all. The Certus complements other pieces of lab automation, whether that be tip-based, whether that be something that can run huge, really fast, whether that's with a washing capability as well. So what it does is it sits in the middle of a whole load of choices that allow us to put together the right automation strategy to best provide for the assay in question. So, for example, where we are doing a number of large screens with qPCR, we've been able to leverage the fact that the Certus has very low dead volumes to dispense out quite gloopy and quite expensive reagents, to allow us to then do the bulk dispensing piece with some of those qPCR reagents on the Certus. Then we'll then take it over to a tip-based system to then do the addition of the samples that have been generated from the cells. So it's a whole bunch of different approaches, and it's about pulling it all together. And obviously the next step from that is then incorporating it into a larger, fully automated piece with arms and scheduling software. So that's the direction of travel with this all as well. And we focus today on in vitro biology because that's where both of us sit within the team, although we may be sort of saying that Ansophy's more focused on hit ID, and I'd be maybe a bit more focused on sort of the hit to lead optimization into candidate selection type areas of biology. It's all very much the same ideas and same philosophies, and we collaborate all the time. And it's not just collaboration within biology. We've used the Certus to positively impact projects that were traditionally lied in places like structural biology, AMI, DMPK, compound management. Again, thinking about the sorts of ideas we've discussed around being able to do more complex things with a lower barrier to entry, because then that allows us to generate good data, allows us to use that data to drive our decisions and hopefully find out more about what we're looking at. And obviously one of the things we're looking forward to is some of the new innovations and new pieces with the Certus, and we've seen lots of exciting pieces in the last few years with the different heads, and obviously talking with the tech team all the time. And we've had a really positive relationship with Sarah and the rest of the team, and we look forward to that continuing. So just with that, a few acknowledgements. I see some people on the call that are on the list as well, but Geiger and Nano, Sarah, Sabrina, and Boris in particular have been really helpful as we brought this into CRL just over 2 years ago now. Had positive impact, and the most important people have been particularly some of the more junior scientists on that list, who have been really open to trying new things and pushing boundaries. And of course our lab management, stores, facilities teams, because without them we don't get to do the fun science we do otherwise. So with that, a huge thank you to everybody for either attending live or for watching this on catch up. If you've got any questions now, we're more than happy to answer them. And then obviously there's some contact details if you've got any questions in the aftermath, or for those that are watching in the future. So yeah, thank you very much for your time, and I'll open the floor to some questions.
Thank you, Gareth. Thank you, Anne-Sophie. That was such a well presented webinar for us today. And I like how you summarised at the end how the Certus Flex, complements all the other liquid handling and automation out there. And just by listening to your presentation today, it's just shown me, how you're using it to its full capacity, which is great. We do have a few questions. Let's have a look. Can you describe, so you talked about how the Certus is configurable using different sources such as syringes and bottle kits. Can you describe how quickly and easily this process was for you as a user, and how do you decide on the valve types that you're using?
So in terms of, I'll take the valve bit first. So I try and use the biggest valve I can get away with, because my philosophy there is that it's then less likely to clog up. So if I think about what's the minimum volume, what volume am I looking to dispense? So if it's sub If it's less than half a microliter, then I'll probably go to the 0.15s. If it's half a microliter, the 0.2s that's pushing it, but that's pretty good, and that's where that data for the 1.6% CV came from. It's thinking about what is the solution? What's my minimum volume dispense? How many wells and what sort of timescales? So obviously the bigger valves, you can push through things a bit faster. They're slightly less likely to clog if you've got things like beads or cells. One of the things we are starting to think about is, well, what about the size? We're thinking about cells, thinking about the size of the cell as well, making sure that we're not going to sort of disrupt things and destroy the cells as they're being pushed through the valves as well. In terms of different sources and syringes, a little bit of that is thinking about what the dead volume is, how much of that reagent can I spare, how expensive is it? If we're thinking that something's really expensive or really, really precious, we're going to be thinking syringes, direct. If we've got it in bulk, I'm probably going into the tubes or the bottles, just on the basis that it's just a little bit easy to switch things over and clean it at the end. Having to play catch with reagents underneath with the syringe is a little bit tricky, and also just the top of the configuration, thinking about how it's all lined up. Having 4 or 5 syringes on top is very doable, but it is. You've then got to think about just can be quite simple. That's where there's more likely to be sort of user error. Well, putting into that one or that one or that one, it can get quite congested. So it's a little bit of how you think about it as well. I'm never going to tell somebody this is the way, how they work in the plate, how they design their assays, but those are the sorts of things we're always considering when we're thinking about which one. And it's a little bit of, you just got to kind of suck it and see really at times and just see what works.
Good. You mentioned the cleaning station quite a lot. What cleaning reagents do you actually put in the bottles? What are you cleaning the valves with? Is it just hot water, ethanol hot water, or do you have other cleaning reagents?
Yeah, we do have 2 protocols on the valve cleaning station. So we have one which is like water and IMS quite a lot. Indeed, we really wash them thoroughly. But we have another one with NaOH also, which is combined with water. Yes, and that really helped to clean really deeply. Let's say,
Yeah, we think a little bit about making those are sort of the basic ones. If we have anything that would be particularly more challenging, we could reconsider and think about different solutions,
like Tergazyme or something like that.
Yes,
we use that. Yes.
So, but we think we just the advantage of the station as well is that it removes that patience factor when it comes to the end user, because you just press the button and then they can walk away and it gets done. Also, just thing to think about when you're washing through any automation dispense heads is thinking about what's in there as well. So we try and make sure we flush it through with a lot of water first, because we're often dispensing nucleic acids. If we're going to start throwing lots of ethanol and things through it, then it might precipitate them out. So we are thinking about the order in which you do things is equally as important as what you use as well.
Yeah, good. Another question. Did you so we've got the new, I don't know if you've seen it, the magnetic stirring syringe system. Have you tried it? Have you considered using it?
We have not tried it. We have had a look at it at Elrig last year, which is how this all came about. We are thinking about that. I think for the uses we've had at the moment, where we're dispensing things that need to be kept in solution hard, they've tended to be smaller, more routine pieces. So it's a case of add it and just put it on. It is a capability that we are definitely interested in, and thinking about how it would fit with sort of larger high throughput screens and the wider automation strategy we have at Charles River. So yes, it is something that we're interested in. We haven't got direct experience, but like some of the other pieces that we've sort of hinted at, we always.
Well, you'll be pleased to know I do have a demo, so I will bring it over and I'll leave it with you, and maybe the next webinar can be about that. We've got somebody you mentioned the new Certus. Where's that question gone? Somebody's asked I think at the end of your presentation, you said that you were looking forward to seeing the redesign. Let me just see if I can share my screen. So, can you see my screen?
Yeah.
Yeah.
Okay, so this is just a quick overview. Somebody asked about the redesign Certus Flex. So this is what it will look like. So we'll have up to 18 channels. There's a comparison sheet which I'll share with everybody in the presentation, and those that registered as well. So if you're interested to know a bit more about this, I won't go through everything, but we do have a brochure that we can share with you. So I know I think you saw. Have you seen this, Gareth?
I think we
Yeah.
were going over that coffee where this all started at Elridge.
Yes.
Last year. That was kind of the way into the discussions, was having a look at having a nosy and a look at the redesign, and obviously thinking about the power that the increased number of channels could have for some of those DOE type experiments in particular, right? So we've got working with a colleague at the moment, and we're thinking, right, we want to do a buffer optimization. We might end up with 20 different fluids, which is brilliant, because that gives you an incredibly thorough and really interesting experiment in 1536. But that's a lot of different solutions to start moving around. So now that those little bits there that mean that we can do it in maybe 2 presses of the go button rather than 4 or 5. Again, they're solvable problems, but it makes the barrier to entry for the end user a little bit easier.
Yeah. So when it's available again, we'll bring one over, and you can see how it will impact your workflow as well. So that's all coming soon. Let me see if there's any other questions. As we mentioned, this webinar, it has been recorded. Contact details for both Anne-Sophie and Gareth and myself will be available. So if you have any other questions, if you'd like to reach out to us, either of us, just feel free to do that. Again, I'd like to thank Gareth and Sophie for this webinar and for your time today. And to everyone who joined, thank you, and I hope you have a good rest of the day. Thank you so much.
Thank you very much.
Thank you.
Thank you. Bye-bye.
Bye.